Can you all see the word file? Muco Polysaccharides. They are slimy substances formed from galactose, mannose, uronic acid, mucopolysaccharides are found both in mucopolysaccharides are found. I have a question for you here. I want to see who will answer this. Mucopolysaccharides are found. Let us write down and then I'll ask you a question. In both plants and animals.
Ma'am, what is written after mannose?
Uronic acid. Done? A question for you all here is, tell me, or have you studied mucopolysaccharide anywhere? Have you studied an example for the mucopolysaccharide anywhere? And where have you studied? Who can tell me this? Very good. Clap for Yash. Everybody will clap for Yash. But what is the example, Yash, that we studied mucopolysaccharide? In the connective tissue,
Ground substance.
ground substance, yes. What is that ground substance made up of? Mucopolysaccharide. Which is an example we studied in the ground substance? Yes, can you tell me? Understood? Have you heard this? I told you hyaluronic acid is a mucopolysaccharide, right? Let me check the answers. Lisa, very good. Heparin, very good. Heparin is also a mucopolysaccharide. Clap for Lisa also. Very good, Sanika. It is hyaluronic acid. Next. Next, we'll be studying about the protein. Okay. Proteins. Let's study the proteins. Essential elements in proteins are carbon, hydrogen, oxygen, nitrogen. Carbon, hydrogen, oxygen, nitrogen are the essential elements of proteins. Write down. After water, protein is the most abundant compound in the protoplasm. You know what is protoplasm? Cytoplasm. Mostly the water content is more in cytoplasm, but there is a presence of, after water, it's a protein which is present more. You all know proteins are the polymer of amino acids. So, if example, this is A protein. How is this A protein formed? A protein is formed by linking different amino acid. This is one amino acid. This is one amino acid. This is one amino acid. So one amino acid, two amino acid, three amino acid, four amino acid. Combination of different amino acid will give you a different proteins. Remember, proteins are always heteropolymer. They do not have homopolymer protein. They are heteropolymer because they have only 20 standard amino acids. How many amino acids? How many letters do we all have in the alphabets? English alphabets?
26.
26. How many names you can form out of 26? How many names you can form out of 26?
Infinite.
Huh?
Infinite.
Infinite. Very good. Same way, you have 20 amino acid, but the combination of this can give to n number of proteins. Did you got the point? What I mean? Collagen is the most abundant animal protein, and rubisco is the most abundant plant protein. You have to know both the proteins. One is collagen, other is rubisco. Collagen is animal protein and rubisco is plant protein. Are you understanding? Write down each and every line. If you have not understood anything out of this, you can ask me. Okay. Any doubts to anyone?
Ma'am,
Yeah, tell me.
ma'am, how proteins are both polymer and heteropolymer?
Polymer means what? Chain. Okay. Heteropolymer means what? Made up of different amino acid. That's it. Hetero means different. Polymer is chain. They are the polymers of amino acid. But what kind of amino acid? What do you mean by homo or hetero? Homo means similar. एक ही.
Yeah.
For example, this is A amino acid. So it is made up of only A chain? No. It is made up of A amino acid, B amino acid, then C amino acid, then again A amino acid. This will give rise to protein. So they are said to be heteropolymer.
So overall, we can write they are heteropolymer.
Overall, no. The question will be proteins are the polymer of dash. So the answer should be amino acid. You cannot wait for heteropolymer there.
Okay, ma'am. Got it.
Mostly question is asked as a polymer of. Proteins are the polymer of amino acid. Carbohydrates are the polymer of. Lipids are the polymer of. Are you understanding this kind of questions? Animal protein is collagen and plant protein is rubisco. Yes. Yeah. Done. Next. Amino acid are group of organic compound having two functional group, and one carboxylic group. There is one amino group, other is carboxylic group. Okay. Amino acids are a group of organic compound having two functional group. Which is the two functional group? One is amine group, other is carboxylic group. Amine group is basic, whereas carboxylic group is acidic. R can be H. This R is a side chain. R can be H. If R is replaced in this structure, okay, in this structure from here till here, are you understanding? This cannot be replaced. This is the structure of amino acid. Amino acid is having amine group and carboxylic group. I'll give you time to write down. Do not write. Understand it first. I'll give you time to write it down. Everybody is going to listen. Drop the pen and listen to me. This is amine group. This is a carboxylic group. Two functional groups are present. But this is not changed in any of the amino acid. Only R is replaced. Only R is changed in each amino acid. If R occupies by H, it forms glycine, amino acid. If R is occupied by CH3, it forms aniline. If R is occupied by hydroxymethyl, it becomes serine. In others, it can be hydrocarbon chain or cyclic chain. So whatever the R is occupied, a different form of amino acid is formed because there are 20 different forms of amino acid. The 20 different forms of amino acid will differ only at the region of what? All the 20 amino acids differ in the region of what? R. Very good. All amino acid contains carbon, hydrogen, oxygen and nitrogen, but some of them additionally they have, yeah, glycine is the simplest. Some of them additional will have, yes, called that is sulfur. Those are sulfur amino acid. We'll study in detail. Physical and chemical properties of amino acid are due to amino, carboxylic and R functional group. Write down this. Now you write down, I'll explain you again. Done?
Just a minute.
Yeah, no problem. Write down. Take your time, understand it, and then write. Understand. Very important. No problem. Take your time. Write. Done, Rishikesh? Is it done, Rishikesh? Okay, write down, write down. Let me know once you're done. Others, I'm hoping that you are done.
Done.
Done? Okay, fine. Yeah. Now, see, the carbon which is carrying both amino group and the carboxylic group is called, this is called alpha carbon. The carbon amino acid or the carbon that carrying amino group as well as carboxylic group is, this carbon is called alpha carbon. Everything will remain same. This everything will remain same in all amino acid. Only R is getting replaced. Only R is getting replaced. So you get 20 different types of amino acid. Who didn't understood this? Carbon, hydrogen, oxygen, nitrogen is present in protein. What is present in carbohydrate? Who will answer? In protein, you have carbon, hydrogen, oxygen, nitrogen. So what is present in carbohydrate? In protein, it is carbon, hydrogen, oxygen, nitrogen. So what is present in carbohydrate? Who can answer? Amod, very good. Carbon, hydrogen, oxygen. Yes, where is oxygen? Hydrates of carbon, carbohydrates. Hydrates of carbon, yes. It's carbon, hydrogen, oxygen. Clear to all? Okay, next. Next, there are different types of amino acids. The first one is neutral. The first one is neutral. Neutral amino acids are, listen to me, neutral means there is one. Here you have one amine group and here you have one carboxylic group. So this type of amino acids are called neutral. One amine group, one carboxyl group. So that is what this says. Neutral amino acid. This is a classification of amino acid. Okay. One amino acid with one amino and one carboxylic group is called neutral. Very, very important table, my dear students. I hope you remember there was a question from here for this NIT. What was the question? Who can answer me? What was the question? Okay, you don't remember? I'll tell you. So, what do you mean neutral? Neutral means they have one amino and one carboxylic group. So, which are the amino acids that are neutral? Glycine, alanine, valine, leucine, isoleucine. Next, acidic means they have one amine group but one extra two carboxylic group. This is acidic. Example, aspartic acid, asparginin, glutamic and glutamine. Basic. They have two amine group and one carboxylic group. Basic. There are two amine group. So arginine, lysine. Now you remember what was the question? Which among this is a basic amino acid? Yes or no? Do you remember the question? Which among this is a basic amino acid? Is this question was there? Are you all there? Listening to me? Hello.
Yes, ma'am.
Hello. So tell me, do you remember which
Yes,
was the amino
ma'am.
acid? Lysine was an amino acid that was basic. Same way you might get question this year, which among this is a neutral or which among this is a sulfur containing, which among this amino acid is aromatic, which among this is an acidic amino acid. So you should know them. Okay. Now, yes, containing amino acids, the amino acids having sulfur.
Ma'am, we have to write this.
Very, very important. You have to write this. I'll tell you. I'll give you time to write. You understand this first. Amino acid containing sulfur are called sulfur amino acid. They are cysteine and methionine. Alcoholic. Amino acid having OH group are called alcoholic amino acids. Structure you do not have to know, but the names you need to know. Serine, threonine, and tyrosine. Aromatic. You know aromatic chain? Something like this, right? Yes, aromatic chain. You draw like this aromatic chain, right? So if the amino acid is containing aromatic chain, then those are called aromatic amino acid. Phenylalanine and tryptophan have aromatic chain. Then heterocyclic. In heterocyclic, you have hetero means heterocyclic. Here you have amino acid having nitrogen in the ring. Here you have nitrogen. Rest everywhere there is carbon. So if they have nitrogen in the ring, they are said to be heterocycline. and this is found in histidine and proline. All this is important. Quickly write it down. We'll learn the trick also. Done? Any doubts? Okay, last line, last line, write down. Okay, others, quickly revise. I'll be asking questions. Neutral, today itself, you are going to revise this. Today itself, you are going to learn it. Neutral, acidic, basic, sulfur containing. Many a time, they'll ask you, I'll tell you which are the ones they ask you. They have asked basic this year, you know. They ask sulfur containing, aromatic, heterocycline. Most of the times, okay, everything is asked, but most of the times, these questions are asked.
Excuse me, ma'am.
Yes.
In acidic, you said it is one extra carboxyl group, no?
Yeah.
So it can be two amine and three
Three.
carboxyl.
Yeah. Yes. Yes. One extra. Then the amine group. Okay. Done. Rishikesh, last line is over. What about others?
Yes, ma'am.
Okay.
Done, ma'am.
Okay, fine. Others? Sakshi, Shifa, Amal, Ishan, Shipra, Chitra, Madhura, Vaibhavi, Sanya, Raghavi, Zenobia, Mangesh, Is it clear? Have you all understood until here? Can we move to the next point if everything is clear? Okay.
Yes, ma'am.
Okay, fine. Next, next write down properties of amino acid. Very important. This is very easy. I'll explain you. Properties of amino acids. One of the most important property of amino acid is, write down, one of, this is amino acid, okay? Amino acid. Amino acid short form is AA dash. One of the most important properties of amino acid is it exists has zwitter ion. Important. Exist has zwitter ion. What do you mean by zwitter ion? We'll draw the structure and then we'll discuss. CH, COH, carboxylic, NH2, and then that is R, right? Now, what happens? It forms CH, COO, NH3 plus and R. So you have minus, here you have plus. This in aqueous solution. So in aqueous solution, in an aqueous solution, amino group gain H and becomes positive, whereas carboxylic group and carboxylic group release H and form negative. So that dual ions formed. And this is known as zwitter ion. Formed an amino acid having both positive and negative, hence known as zwitter ion. I hope you understood. If you have not understood, you can ask me. Is everybody done?
Yes, ma'am.
Okay. Anybody, any doubts? Next. Nutritional classification of amino acid. Okay. It is first one is essential amino acid. Essential amino acid. Amino acid, write down, amino acid which cannot be synthesized, in the body are called essential amino acid. These are 10 for children and 8 for adult. First one is phenylalanine. Second, valine. Third, tryptophan.
Please follow.
Okay, write down. Let me know once you're done.
Scroll down.
Tryptophan, then threonine, isoleucine, methionine, histidine, arginine, phenylalanine. Alanine, it is not mine. Leucine and lysine. So how do you remember this? A trick to remember. Listen to the trick. Trick to remember. P V T T I M then H A L L. So you have one is Hall, Other is Tim, and then is this private Tim Hall. Understood? Easy way to remember? Private Tim Hall. Whereas histidine and arginine are synthesized only in the adults. So, tryptophan. I'm repeating: phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, lysine. Private Tim Hall. You need to know the order. Then you can easily understand well. Done? Write it down. Private Tim Hall. Done? Hello. Are you all there? Is it
Done.
Okay, fine. Now we are studying the second one as non-essential amino acid. What are non-essential amino acid? Which are synthesized in the body. The rest all are coming under non-essential amino acids, such as, we'll write down, glycine, cysteine, alanine, serine, aspartic acid, glutamic acid, asparagine, glutamine, tyrosine, proline. Now I'll give you time. You all will study the protein part and do let me know if you have any difficulty. Quickly, everybody, everyone, 50 of them or 50 plus of them are going to study the protein part until here before we move to the next. Quickly. No tricks for this. You, Rishikesh, you find out trick for this and get back to me. Clear? I don't have trick for this. So anybody can find out trick for this and get back to me. Quickly learn. Next, we need to talk about protein structure. So quickly learn. Everybody is going to learn there. No wasting time. Important. See, this year also there was a question. There was a question from carbohydrate. There was a question from protein. This chapter is important, right? 8 marks, completely 8 marks. Easy questions. That too very easy questions.
Ma'am, which is the fifth acid in non-essential amino acids?
Aspartic acid.
Ma'am, could you please
Aspartic.
pronounce it again?
ASPARTIC.
Okay, ma'am.
I'll ask you questions. Quickly revise. Can we move to the next? Any doubts to anyone? Hello. Any doubts to anyone?
No, ma'am.
Is it clear to each and everyone?
Yes, ma'am.
Yes,
Okay, no doubts? Well, good. No doubts? Very nice. Now let's move to the structural part of protein. Okay. Structure of protein. Four basic structural levels are primary structure, secondary structure, and tertiary structure. Okay. First, we'll discuss about the primary structures. Primary structure here, it is a linear specific sequence of amino acid. Linear, straight chain, which is made up of different amino acids. The bond between them is what kind of bond do they have? Who will answer? Between the two amino acid, what kind of bond? Peptide bond.
Peptide.
One end of the chain is N-terminal, other end of the chain is C-terminal. The first amino acid is known as N-terminal. This last amino acid is known as C-terminal. Simple. Primary is very simple structure. Right. Very easy. First is primary structure. You can draw the diagram. It will be much clearer for your understanding. जी बेटा. Are you done? Okay. Next. Secondary structure. Secondary structure is formed. See, primary structure is a single, right? Secondary structure is formed by coiling. Secondary structure is formed by coiling. Is it clear? However, the coil takes place whether it is in helix, whether it is in stepwise, or whether it is coiled. So, those are leading to secondary structure. So, primary structure is what? Linear. Very good. Now we are talking about the secondary structure. It is formed by the folding of primary structure. Okay, that is secondary structures. So here you have alpha helix, beta pleated, and collagen helix. You can get three forms where it can be helical structure, you can see, or it can be beta pleated structure, or three linear polypeptide can twist to form collagen helix. Examples for alpha helix, please write down. Examples for alpha helix is keratin protein. Keratin protein present in hair nail. Beta pleated examples are, write down, keratin protein or beta keratin. This is alpha keratin, okay? Beta keratin present in scales, feathers, scales, feathers. Very important. Fibrion is what? Natural silk. Protein present in silk is beta carotene. And triple helix where three polypeptides are twisted around, it's found in collagen. Collagen protein. Write down. Okay, write down the diagrams also. You do not have to know the inner structure, but yes, you have to at least make a note of this diagram. If the question comes for at least 2 or 3 marks, you'll be able to write and you'll be able to draw it and explain them. Done? Are you done?
Yes, ma'am.
Next, tertiary structure. Tertiary structure. First, okay, if you're done with the secondary structure, let's move to the tertiary structure. Tertiary structure is formed by the folding of secondary structure. Okay, formed by folding of, sorry, folding, not, is it? By folding of secondary structure. Primary is formed by the folding of, sorry, secondary is formed by the folding of primary, and tertiary is formed by the folding of secondary structures, right? They can fold among themselves. They can fold among themselves upon itself, forming a ball-like structure. These tertiary structures are basically found in enzymes and myoglobin. You know what is myoglobin? You know, enzymes, we always say to the function for the enzyme to be proper, they should be in their particular structure, tertiary structure. If there's increase in pH, if there's increase in temperature, then the enzyme become inactive. Why we say the enzyme become inactive? Because they lose their structure. The enzyme, the proteins, the polypeptide, if this is an enzyme, okay, trypsin is, this is a trypsin enzyme. If this trypsin enzyme, if doesn't fold like this, then it is not active. So if it becomes from folding, it becomes to the linear, it means it is inactive. It is inactive, and this is active. So enzyme, myoglobin is a, what is myoglobin?
Oxygen binding protein.
Where it is present?
Skeletal muscle tissue. Skeletal
Muscles.
muscle.
Very good. Very good. Very good. How many oxygen does they carry?
1.
1. Very good. Then quaternary. Quaternary structures. What are quaternary structures? Protein are said to be quaternary structure if there are 2 or more polypeptide. If a protein is having 2 or more polypeptide, it is said to be quaternary. So can you tell me, give an example? That's an example is already given. It's a hemoglobin. Hemoglobin is made up of how many polypeptide chains? 4. 2 alpha and 2 beta. You know that. So this is called quaternary structures. Write down. Everybody write down. Any doubts to anybody? We are done with the structural part of proteins. Does anybody having any doubts? Is everything clear to all? See, if you everything clear, then only you tell me yes. If not, you can ask me. You can clear your doubts. Do not leave your teacher until your doubts are clear. Everything clear for today's class? So, should we wind up for today? All the 60 plus students, is it clear?
Yes,
This
ma'am.
question is not for one or two, it's for each and every person who is attending the class here. Okay, if it's clear, it's well good. So, we'll be discussing the rest in the next class. But I want you all to go through carbohydrate and a protein once again. I have given you ample of time today to go through it, right? So I want you all to go through it again before we start or before we continue with this. Is it clear?
Yes, ma'am.
Okay. Everybody will do that? Okay. I'll sign off for today's class. See you all in the next class.
-2 आणि b मध्ये 1 आणि c मध्ये -1 ह्याच्या आन्सर क्लिअर ab प्लस a 1 ca मायनस bc 7 ab
Keep learning and bye-bye. Take care.
Thank you, ma'am.
Thank you, ma'am.
Welcome. Bye.
Thank you,
Take
ma'am.
care. Welcome. Bye-bye.